Good morning, everybody. And today I'm going to do one video just to explain some questions that you had on the previous activities, and I'm going to introduce a new technique, a new important process in biotechnology. Well, first of all, this question that a lot of you had wrong in the activities that I asked you to do last week, and I didn't consider that such a difficult activity, but now I'm sure that I didn't explain it in the right way. Well, the first thing we're going to see is that this is not a real DNA molecule, as it only has one of the strands, but it's just to make it easier for you to do the activity. And it says that we've got this DNA and we are going to cut it with this enzyme. And we've got this information, we always need this information. What is the sequence that this restriction enzyme recognizes and therefore cuts? In this case, this sequence recognizes this enzyme recognizes this sequence CTTAAG. So will it cut this molecule? Yes, it will, because we've got here those 6 letters CTTAAG. No doubt it will cut it. But the question now is, if it cuts this DNA molecule, what will be the way it looks after cutting it? Well, we've got another information that it always cuts between A and G. Obviously, A and G in this sequence, because the enzyme is not going to read anything else. So I've got to go and see this sequence and then see A and G, and the restriction enzyme will only cut there. So what's the right answer? What's the only possibility? This one, letter C. I've got the whole DNA molecule and then I've got it, cut it there. This was the right answer, letter C. Of course, there's more A and G in this DNA molecule. And some of you said that these could be possible as well, but it's not, because it must be A and G in the sequence that the restriction enzyme is recognizing, okay? Well, there's another activity here that some of you had problems with. We've got different patterns that different enzymes read, and we've got 4 examples of different DNA molecules. So what do we have to see? Well, the only thing we're going to do is, this is going to be our DNA sequence, the first one. Well, I'm going to look in this DNA sequence things that call my attention. For example, I've got 2 double G there and 2 C's there. So is any enzyme going to recognize something similar than that? Well, no double G there, but is that GGAT? Is it like that in my molecule? GGO, and I have AT there, and then a double C. So where should this DNA molecule be located? It should be located there in the second box. So you just had to write a number 1 there, as this is the molecule that was going to be cut by that enzyme. Okay, well, we're going to see number 2. Do you see any particular thing in this molecule? Well, we've got a double G there, and very close another double C. Well, it cannot be this one, but we've got that one there with a double G. Is it double G, T, T, C, C? That's right. So this enzyme will cut this molecule. It will not cut this one, and it will not cut any of those molecules either. Only this one that contains this sequence. And where will it cut it? Between G and G. Okay, we've got G and G there, G and G there. That will be the way this one will be cut. So this will be number 1, and that will be number 2. And then we've got number 3 and number 4. Where should we put each one? Well, we've got number 3 here. If you read it, we've got double A, double T. Anyone that is recognizing that one? GAATT, and then a C. GAATT, and then a C. So number 3 is the one that is recognized by this enzyme. And then number 4 double A, G, C, T, T, anything that that you see there, well, it's the only one that is missing. Okay, so we're going to have number 1 there, number 2 here, number 3, and number 4 with this double A, G, C, T, T there. Okay, so that was activity number 4. The only thing you had to do was just locate the right number in the right box. And then on the last video, I showed you this picture, and this picture was showing the difference between those enzymes that when they cut the DNA molecule, they leave sticky ends, and those that they don't. What's the mission of this sticky end? I told you that it's very important to have a sticky end whenever you want to join this DNA that you've cut with another DNA molecule, the DNA molecule from another organism. So in this case, it's very useful to have a sticky end. If you're not going to join it with it, if you're just going to analyze the DNA that you've got, sometimes it's enough to have an enzyme that cuts the DNA like that. So we've got sticky ends and blunt ends. That was one of the questions in the questionnaire. It was just asking you which of them produced those sticky ends. Which of these? Well, all that have this pattern, not a straight cut. This pattern, that one, that one, that one, and that one. So you only had those 3 examples there, but of course this one, I3, that's not a sticky end, and that one is not a sticky end either. Okay, so now you know the difference between sticky end and blunt ends. And this question was very easy as well. It was just asking you to read this whole DNA sequence and which of these enzymes will cut it. Well, again, we go and see special things that we've got, and then we're going to see that GTCG, AC is only present in, is only recognized by these enzymes. So that's the one that is the right one. Remember, these type of activities just need your attention. As long as you pay attention, it's not going to be difficult for you. That's the way that this restriction enzyme will cut these. And I just wanted you to realize that every section that a restriction enzyme recognizes is always symmetrical. So if you read it from this side, it's GTC, GAC. But if you read it from this side, GTC, GAC, it's always like that. Okay, you're going to see that it's always like that. That's because this one is the complementary base of that one. This one is the complementary base of that one, and that one is the complementary base of that one. So if you take the axes, they are always complementary one to another. So you can read them in both sides. Just one particular thing, that every area that a restriction enzyme recognizes is going to have this particular characteristic. Okay, so that's another enzyme that is going to leave sticky ends, those nitrogenous, those nucleotides that want to have a complementary one. So all restriction enzyme recognized in recognition sites, they are all the same forward and backwards. That's what I meant. Okay, just take the one that you prefer, TGG-CCA, and we've got TGG-CCA, and that's because if you take the axis, this one is the complementary base of that one, and we have the same here if you take the axis ATCG, so we read them forward and backwards with no problem. Okay, another question that caused you some problems, that's because you didn't do the picture. I'm sure that if you make the picture you're going to see this in a very easy way. We've got a circular piece of DNA. What does it mean that the piece of DNA is circular? Well, it means that it's bacterial. Okay, remember, bacterial DNA has this shape. And we've got 3 sites for a particular restriction enzyme. So it's going to have 3 places all along this DNA molecule, 3 places with the sequence that this restriction enzyme recognize. So 1, 2, 3. And then the question is, how many fragments will this restriction enzyme how many fragments will that restriction enzyme cut the DNA? So we've got 1, 2, and 3. Can I know the answer? Could it be predicted? Yes, always 3. But it's different from the next question, that we've got a linear piece of DNA. That's our human DNA. Any other eukaryotic organism DNA that has a linear DNA. Then if this particular restriction enzyme cuts in 3 places, is it going to have 3 sections? No. In this case, it will be 1, 2, 3, and 4 fragments of DNA. That's the way it works. So it depends whether the DNA molecule is linear or it's circular.